So there's another hurdle to this technology. Even if you say, okay, I'm going to allow this technology because it's important to come up with new possible cures for regenerative medicine, there's still another hurdle to the technology, and that hurdle is that the immune system recognizes foreign cells and it eliminates them from your body. So if I give you embryonic stem cells from a fertilized egg generated in the fashion that I just described to you, and you take those embryonic stem cells, which we perhaps can differentiate into a particular cell type, such as a pancreatic islet cell, if we put those cells into your pancreas, your pancreatic cells and your immune system are going to recognize that that is a foreign cell, and your immune system is going to wipe that foreign cell out. So given the technology, there's still a problem, even if you say that you will accept the ethical concerns involved and that the moral concerns of trying to advance this technology for the use of curing various different types of diseases and degenerative disorders is worth the research involved. So a pancreatic islet cell then, made from an embryonic stem cell of an unmatched embryo, is still going to be rejected by the body. So what other methods can be used to generate stem cells with broad potential for regenerative medicine? Well, scientists have thought about this a lot. In fact, we've worked on this a lot. And one technology that has been developing over the last 5 to 10 years is that of nuclear transfer research. This is the technology that's often mistakenly referred to as human cloning. The technology that was used to create Dolly, which was the first cloned animal, happened to be a sheep, which was made in England some years back. So this technology has the capacity to generate more and more and more of perfectly genetically identical animals. So what is this technology involved, and how can it be useful in regenerative medicine? So let me just point out from the very beginning that scientists and society are opposed to human cloning. So what is this technology then? How can we use this for regenerative medicine? How can we use it in an ethical manner? So let me first describe to you the process of nuclear transfer. In this process, what we do is to take an unfertilized oocyte. And my laboratory does this type of technology. We work on mice to be able to understand the process better. So what we do is we start with an unfertilized oocyte, a single cell coming from a female, and we then remove and discard the nucleus of this oocyte. So this is effectively an oocyte now, but it has no genetic information. So now we give it some genetic information. We take and replace it with the nucleus of an adult stem cell or any other type of somatic cell. So we could take a small piece of mice skin, pull out the nucleus from that cell, and implant it into an unfertilized oocyte. We do that process from mice. So what do we do next? So we now have generated a hybrid cell. The cytoplasm of the cell comes from the unfertilized oocyte. The nucleus of this cell comes now from an adult somatic cell. This is a diploid nucleus because all of the cells of your body, the somatic cell is basically diploid. So even though this started out as an unfertilized oocyte with only the cells from the female, basically what the cell now has is a diploid adult nucleus sitting in a oocyte cytoplasm. So what good is this cell now? Well, what this cell can now be used for is we first take this cell, we put these cells in culture, and again, I work on the mice, take these cells, we put these cells in culture just as I described to you on the previous slides. We generate embryonic stem cells, just as I described to you from the previous slide. We can generate blastocysts, many blastocysts, just as we described. Again, everything in vitro at that point. But then we take the blastocysts, and now we take those blastocysts, and we put those blastocysts into a female host. And what that female does is 3 weeks later produces pups, mice. And some of those mice, not all those mice, but some of those mice are healthy. And those mice then continue to grow to adult age. And in fact, these mice now are several years old, and these mice are perfectly healthy, and we cannot tell the difference between these mice and a normal mouse. And yet these mice came from an oocyte with a diploid somatic cell, and that somatic cell came from a skin stem cell, the type of cells that my laboratory works on. So these healthy mice then are great. The problem is that there's still a problem with efficiency. And in fact, even though we've got a couple healthy mice, in fact we have more than a couple healthy mice, the problem is that even on the best of days, there are still only about 5% of all of the blastocysts that end up surviving to this point. And there are many embryos that do not develop properly. So this technology is obviously not perfect, and there are problems with taking an adult nucleus and putting it in the oocyte cytoplasm, even though those hybrids, in some cases, can give rise to normal, healthy mice. But now consider the situation for humans. I already told you that we as scientists are completely against the notion of human cloning. So we don't want to get to this stage. The only stage that we want to get to is a stage of generating human embryonic stem cells from nuclear transfer. So what ideally we would like to do is to take cytoplasm from a human oocyte and put into that cytoplasm a nucleus from your skin cell, for instance, if in fact you are the one that is going to be in need of regenerative medicine. And from that, we then want to culture those cells to produce embryonic stem cells. But now, so now we take the exact same step that I just described to you, the nuclear transfer experiment, and we now put the cells into culture, just as I described to you earlier, culture them to the hybrid cell stage to form a blastocyst. And then the third step is derive our embryonic stem cells from the blastocyst. And now the benefit is that now we have embryonic stem cells that perfectly match the patient's DNA. And that means that if we now generate pancreatic islet cells from those embryonic stem cells that are perfectly genetically matched, now the patient's immune system is going to recognize those cells. Even though they came from a hybrid, the genetic content was in fact the patient's genetic content. And so this cell basically is not going to be rejected by the immune cell. And so by generating embryonic stem cells from a skin stem cell and making neurons or muscle cells in culture, the resulting cells are not recognized as foreign by your immune system. So right now, scientists have potentially gotten around an enormous hurdle that just a few years back presented a serious problem. But now what are the problems remaining? Well, unfortunately, there's still the problem today of generating healthy human embryonic stem cells from human oocytes and somatic cell nuclei. It works quite well, in fact quite beautifully, for mice, even though the efficiency of producing healthy cloned mice is only about 5%, 2%, 1%, depending upon the various different researchers and the various different cell types involved. In fact, it turns out that to date, there's very little evidence, if any evidence, that in fact we can adapt this type of technology to humans. That said, I think scientists feel very optimistic that it's just a matter of learning more about differences between mouse embryonic stem cells and human embryonic stem cells, and a matter of learning more about how to improve this type of technology and how to adapt technology that we can already use for the mouse and adapt that to human.